Welcome friends, in this video we are going to look at the question 5 of biology paper 2 for the year 2023, and the question reads: a hemophilic mother had two sons who were both hemophilic. The father was non-hemophilic. Question 8, define the term hemophilia. So the first thing that we are going to look at before we answer the questions, first let's share one or two about genetics. So genetics is just the branch of biology that tells us how characteristics are transferred from the parents to the offspring. So we need to have the parents, then the characteristics must be transferred to the offspring. Okay, so now the characteristics that we are looking at here are things like eye color, the color of your hair, then skin color, sometimes even height. So these are characteristics that we are looking at. Now, if you look at the word genetics, we can divide it. If we remove TIC, we have the word gene. So gene is just the basic unit of genetics that tells us how this inheritance is done. Or in a gene is where the characteristics are being carried as they are transferred from the parents to the offspring. Now, for you to understand fully about genetics, first you need to look at reproduction, because reproduction tells us how living organism produces their young ones. So during the reproduction is when the characteristics are transferred. Now, after we have some ideas about genetics, and if you look at the word generation, from the generation we have gene. Thus, we have the transfer of characteristics from one generation to another generation. So the whole thing is focused on a gene. Now, if we are to analyze the cell, we have the cell here. Then inside the cell, we have the nucleus. And the nucleus controls the metabolic reaction that occurs in a cell. Then inside the nucleus are chromosomes. So we have chromosomes. Then chromosomes will give us genes. So the genes are found in the chromosomes. Then in the genes we have the proteins such as DNA. Then inside the DNA is where we have the process of heredity. So heredity tells us how the characteristics are transferred. Now, away from there, since we have at least a sequence how this is done, let's also look at the chromosome. So how is the structure of a chromosome, and why do we focus on chromosome? Because as we can look at the question, we may have questions that has to do with the information about chromosomes. So the first one, a complete chromosome is made up of two strands called chromatids. So if this is the X chromosome, like the one that we have, we need to have two chromatids. So these are what we call the chromatids. Two strands. Now, the point where the two chromatids join in order to form a complete chromosome, this is what we call a centromere. Now, the chromatids that are found on the same chromosome are known as sister chromatids. So remember some of these terminologies, they are very important. Now, after we have about an idea of the structure of a chromosome, then let's say how many chromosomes do we have in organisms such as human beings. So under human beings, we know that a normal human beings must have 46 chromosomes. Then in this 46 chromosome, 23 chromosome came from a male parent. Then the other 23 came from the female parent. Now, out of these 23, there is one sex chromosome. Okay, so let's say the person that we are dealing here with is a female, and we know that under a female we are going to have XX. Now, out of these 23, there should be 22 autosome plus one X chromosome, even this one, there should be 22 autosomes plus 1 sex chromosome. So if we add 22 plus 22, we get 44. So there are 44 non-sex chromosomes. Then if we add this, we get X. In total, we have 46. So for a woman, if we are trying to like expand for a woman, there are 44 that are responsible for the body and 2 chromosomes that are responsible for the sexy chromosomes. Then for a man, the case is different. We still have 46, yes, so the total must be the same. But out of 23, we are going to have 22 plus X. Then here we are going to have 22 plus Y, because for a man it's XY. Now, if we look at the inheritance of sex-linked characteristics, so these are characteristics that are found in sex chromosomes. Now, if we look at the sex chromosomes, we are just looking at the X and Y chromosomes. Now, the Y chromosomes does not carry the sex-linked characteristics. It's very small. So the only sex chromosome which can carry these characteristics is the X chromosome. Now, let's say let's give a genotype, and we are going to use the same hemophilia. So let's begin with a woman. So for a woman, we know that we are going to have XX. So we'll begin with, if the question says, if the woman is 100% normal, which is okay, what do we expect? So for a woman who is 100% okay, we are going to have capital letter H, even here capital letter H. Then the second one, a woman can be a carrier. So for a person to be a carrier, one must be a capital letter, the other one must be a small letter. Then the last one, for a woman to be a patient, we need 2 small letters for H. So here we have H, also here we have H. So these are what you are supposed to know about a woman. So a woman can be 100% okay, can be a carrier, or can be a patient. So regardless of the letters that you are using, a patient must have small letters, a carrier must have a small letter and a capital letter. Then the normal must have capital letters. In case where you are talking about the red-green color blindness, where you are maybe using capital letter R and small letter r, the concept is still the same. Now let's look at the man. For the man, we only have 2 situations. So if the man is 100% normal, which is okay, we only put the capital letter H on the X. Remember, the Y does not carry the defect. You don't have to put anything to the letter Y. Whether normal, whether sick, the Y does not carry anything. It's small. So for a man, it is either normal or a patient. There is nothing like a carrier, because for a condition to be a carrier, we need a capital letter and a small letter. And we can see the case, since the Y does not carry the defect, we can't have a situation where we have a capital letter and a small letter. So if you are able to have this information about these sex-linked characteristics, then you never fail any question. Now, after we've looked at this, let's look at the examples of sex-linked characteristics. So the first one, we can look at sickle cell anemia. So sickle cell anemia is an example. Then the second one, we can look at the same hemophilia. Then for the third one, we can look at red-green color blindness. Now, each disease here has its effect. For sickle cell anemia, it's a blood disorder that changes the shape of the red blood cells into an S shape. Thus, the name sickle cell anemia. Then for hemophilia, it is just a blood disorder in which blood does not clot normally. It takes time for blood to clot, or sometimes impossible. And the reasons behind this may be lacking of clotting factors such as vitamin K and some ions. Then for red-green color blindness is a situation where a person or a patient with such a disease may find it difficult to differentiate or to see the difference between red and green. So since we have shared one or two, let's go back to our first question. A, define the term hemophilia. So we'll say this is a blood disorder in which blood does not clot normally. So due to, we can say, insufficient clotting factors. Then for B, using letters XH and XH capital letter, write down the genotype of the first one, the father. Now we can go back to the first statement. We are told the father was non-hemophilic, so it means the father was 100% normal, so there was no condition. So the genotype for a father who is okay is this one. Then for the mother, we are told was hemophiliac, so this mother was sick, so we need to use small letters. So we have small letter h, also here small letter h. So that is, so if you're able to master these genotypes that I've given you, then this question will always make sense. Then for question C, using a genetic diagram, explain why both boys were hemophiliac. So let's say parental, or just say parents. So for the parents, let's begin with parental phenotype. So for parental phenotype, let's begin with the man. So the man was normal. Then for the woman was hemophiliac. So for phenotype, under this one, we just have to look at the condition. So the woman is hemophiliac, the man is normal. If we want, we can just say non-hemophiliac. So after parental genotype, we can look at, phenotype, we can look at parental genotype. For the man, it is XHY. Then for the woman, we are saying we are going to put small letters since the case was a patient. Then after that, we need to look at the parental gametes. So for parental gametes, we just have to split. So we have XH, Y, XH, XH. From there, we can now do random. So we have XH. Okay, so this one is just telling us that they had a chance of having a girl who is a carrier. Okay, so another one, we have another carrier. Here, another, so here they have a chance of having a male who is sick. Even here, a male who is sick. So we can tell from the percentage, we can tell that we have, so let's say we do this. So from the ratio, carrier 2, then the patient in terms of the boys also 2. So if we are to find the chance for the carrier, it's 2 over 4. The 4 is the total times 100%. So there is a 50% chance for every fertilization to result in a girl who is a carrier. Then there is also another 50% for a boy to be sick. So with this percentage, we can conclude that there was a high chance. We can see 50-50. So this 50% was the chance that is giving us the picture that there was a high percentage of having a son who is hemophiliac. So let's hope this video will help you to learn one or two. So that's it for this video. Thanks for watching.